Hello, welcome to Wonders of Biology, where I explain interesting biology concepts. Today, I'll be going over Mendelian genetics. So, let's get started. So, with Mendelian genetics, everything kind of revolves around this guy over here, and his name is Gregor Mendel. So, Gregor Mendel was a scientist who wanted to find out how traits are passed along. How do offspring inherit certain traits from their parents? And the way he experimented with this is he used pea plants as his model organism. And there are a few key reasons why pea plants were such a great test organism for Mendel to use and for him to study. So first of all, it was really easy for him to obtain, and there were a lot of strains of pea plants that were true breeding, meaning he knew what traits they would be. Like they have for multiple generations, they had always expressed the same traits, like purple flowers, green seeds, and so forth. And that way he was like confident on what traits that these plants would pass down. Also, the other thing with pea plants is that you can easily control their reproduction. So by taking the pollen from one pea plant and pollinating another pea plant, he could easily just determine who were the parents and what the offspring's traits are and how the offspring was able to inherit the parents' traits. And also, you see all of these little seeds over here. So basically, pea plants, they make so many different seeds in one pea pod. So they make so many different offspring. And through that, you can easily grow so many different offspring from just crossing two pea plants. And that way he could grow a lot of different plants and test and make sure that the model of inheritance that he is seeing is correct. And there's also one more thing. Mendel didn't know this when he selected pea plants, but pea plants actually have a lot of traits that are coded for by one certain gene. So like one gene codes for one trait. And this is really convenient because by testing for like purple flowers, that's only controlled by one gene. So he doesn't need to worry about all of some other complicated nonsense. And he could easily test and see what was going on. So when Mendel began experimenting, he tested how certain different traits in the pea plants are passed down. And one of the traits that he tested for was flower color. So he had these purple flowered plants and he had white flowered pea plants. And he wanted to test basically how does this trait, how is it passed down to offspring? So he basically had some true breeding purple flowered plant varieties, meaning over multiple generations, they are only passing down a purple flowered trait. All of the offspring have purple flowers for multiple generations. And then he also had some true breeding white flower plant varieties. So same case scenario here, they're only passing down that white trait. That's the only trait that is in these plants. So he decided to cross these two plants. So when he took, let's say, the pollen from a purple plant and crossed it with the egg of the white plant, then he came up with only purple flowered offspring. All of the offspring that he got were purple flowered. And this is a little different from the common hypothesis that was held during that time period, what people believed. And what they believed was blending. So they thought that all the traits that were crossed, basically when you have two parents, their traits are sort of blended together. So in this case scenario, you would get like a light purple flower. Maybe like because it's a combination of the white color and the purple color, and you would get that lighter purple color. But that's not what happened here when Mendel tested it out. What he saw was that the white trait seemed to completely disappear, and you only have purple flower traits now. So that kind of confused him. So he wanted to like see where did the white flower trait go. So he decided to cross two more of these plants. And what he saw was most of the times, yes, he did get purple flowers as the trait, but around 1/4 of the time he got white flowers as the passed down trait in the offspring. And so basically you have this reemergence of this initial parent trait coming here again. So what Mendel figured out after experimenting with this and seeing what was happening is that there must be two versions of the trait. And basically, how it's passed down is maybe like there's one trait that codes for purple flowers and one trait that codes for white flowers. And then he also saw that over here, you only have, in this column, you only see that there's only purple flowered plants. So this must mean that the trait that codes for purple flowers dominates that trait that codes for white flowers. It covers it up. So you seeing this, he came up with the law of dominance. So what the law of dominance basically says is that you have one trait that sort of dominates the other. And how we normally denote this when we're talking about two different versions of a gene is with letters. So a capital letter would demonstrate a dominant trait, and a lowercase letter would demonstrate a recessive trait. So over here I'm using P's. So the capital P would basically be that purple flower trait, and the lowercase p would be that white flower trait. So the capital P is dominant, and the lowercase p is the recessive trait. So this is basically what Mendel's law of dominance was. But he also realized that basically what this would mean is that all of these true breeding varieties, they basically have two capital P's because that's the only trait they have. And these white flowered plants, they basically have two lowercase p's because that's the only trait that they have. So that would make all of these plants that you get once you cross these true breeding varieties, they're going to have um, these traits basically. They're going to have one dominant trait and one recessive trait. The word we use for these traits are called alleles. So you would have one dominant allele and one recessive allele. So when you're crossing these, you get this 1/4 of the offspring being white, and that would be because when you cross them, you get like if you have a 50-50 chance of inheriting both of these letters, so maybe like it's like a coin flip. You have a 50-50 chance of inheriting each one of these. So that would give you around a 1/4 chance of inheriting this white color. So because he saw that each parent plant, basically gives one of its traits by random, he was able to come up with the law of segregation. So the law of segregation basically says you have a 50-50 chance of inheriting each trait from each allele from the parent. So that's basically two of Mendel's laws. There's one more law, which I'll be talking about in a few minutes. So basically, with all of that stuff with the alleles and stuff, you can use that to kind of make these Punnett squares. So these are called Punnett squares. And basically, they're a way to kind of predict what traits and what traits and what alleles the offspring will get. So over here, this is kind of like the very first cross. So he crossed the two true breeding varieties. So you have basically the purple one over here and the white one over here. And you can basically write their alleles. So capital P here for purple, capital P here for purple, capital P here for purple, and then the lowercase p's for white. And how these work is basically you just drop down like this, and basically that would make this capital P and lowercase p. And so for the first generation, you have all capital P, lowercase p. So they're all purple flowers. And then for the second generation, when he crossed these capital P little p's, he got that white trait showing up again. So you can kind of predict what the outcome will be in the offspring by using these Punnett squares. So basically what we have here is a slightly more complicated Punnett square. So before we had a Punnett square that only had one trait, which was the flower color. And what we have here is two different traits. So we have our shape and our color of the pea plant seeds. So Mendel, he didn't only just test flower color as his trait, as the only traits which he experimented with. He experimented with a lot of different things in pea plants, and he did it multiple times to kind of confirm if everything he is seeing is true or not. And he saw that the same thing kind of happened, and that's how he like kind of confirmed it and started calling them laws. So over here we have a dihybrid cross, which basically means it's a Punnett square that deals with two traits. So the two traits we are dealing with here, like I said, was going to be our shape. So we have either a round shape or a wrinkled shape, and also our color. So we either have a yellow color or a green color. So basically with this, you can see that these are true breeding varieties, like I said, so they have the same traits, like this. What we like to call true breeding varieties, they have the same trait, right? They both have dominant. So this we like to call homozygous, when they have the same trait. So this would be called homozygous dominant, because it is homozygous for the dominant allele. And over here, we have two lowercase y's, like this. And that would mean it's the recessive trait, right? So this is homozygous recessive because it's homozygous for the recessive trait. So when you cross these, you get your hybrids, right? And your hybrids, they have basically both of the alleles. They have the dominant allele and the recessive allele. So what we call these is heterozygous. So you have your dominant allele and your recessive allele. And you're always going to express your dominant allele because as Mendel's law of dominance says, the dominant allele always masks the recessive allele. So over here, you can see that this is a doubly homozygous dominant seed, and this is a doubly homozygous recessive seed. So when you cross these two, you get a doubly heterozygous seed. So when you're trying to do the Punnett square for this, you can't just put all of this whole thing there, right? It's going to pass down certain traits. So you have your 50-50 thing from your law of segregation, and from using that, you can determine what the gametes that this plant seed is going to pass down. So in order to determine these, we like to use something called the FOIL method. And it's similar to factoring from your math class. So you would take basically the parent's genotype, so we call these traits genotype, and you would basically FOIL them. So if I want to determine the gametes that I would get from this, I would multiply by, it's not really multiplying, but I would pair this dominant Y with the dominant R, and I would get this. And then just like factoring, I would do this, and then I would get capital Y, lowercase r, and then lowercase y, capital R, and lowercase y, lowercase r. So those would be the four different traits that this seed can possibly pass down. So you would put those on both sides of your Punnett square because we're crossing two doubly heterozygous ones. And then you would do a similar thing that you did for your single cross. So you would just basically carry over these traits. So this would be doubly homozygous dominant, and so on and so forth. So when he crossed two doubly heterozygous plants, he came up with this ratio. So he got around a 9:3 to 3:1 ratio. And what this basically shows is that the traits for one of the traits didn't impact the other ones. So the traits that code for the seed's shape did not affect how the plant inherited the traits that code for the seed color. They're both independent of each other. So this gave rise to his law of independent assortment. So this is the third law of Mendelian genetics. It's called independent assortment. Basically, one trait does not impact the inheritance of the other.